Hi everyone! I will be discussing Figure 1 of the research article, The Gut Microbiota Influences Blood-Brain Barrier Permeability in Mice, which contains 7 different parts. Figure 1 is answering the question of whether or not the antibody is able to penetrate the BBB and reach the brain parenchyma. The way the experimenters went about answering this question, or their experimental design, was by administering infrared-labeled immunoglobulin G2B antibody to dams during pregnancy. Figure 1a shows pictures of pathogen-free mouse embryos injected with the antibody at 13.5 days to adult mice. Figure 1b uses a chart to describe the penetration of the antibody into the differently aged mice embryos with pathogen-free mothers. Figure 1c introduces an embryo at 16.5 days of a germ-free mother and displays the antibody presence in that one. D compares pathogen-free and germ-free embryos directly, revealing differences in IgG and CD31 presence. E compares brain regions of 16.5-day-old mouse embryos and reveals the expression of both IgG and CD31 in pathogen and germ-free mothers. F is a Western blot of the brain lysates of 18.5-day-old fetuses with germ-free and pathogen-free mothers, testing for Z01, occludin, claudin 5, and glyceraldehyde phosphate dehydrogenase, which was the control. G is a quantification of the data from the Western blot. Some methods of this experiment were immunofluorescence microscopy and Western blot. Immunofluorescence microscopy is used for locating proteins inside a cell by labeling them with a detectable substance and injecting them into the protein into the organism. Western blot is used to identify a specific protein by gel electrophoresis, separating and illuminating the antibody. A control was used in the Western blot to ensure proper functioning of the method. The control used was glyceraldehyde phosphate dehydrogenase. That pretty much sums up Figure 1 of the experiment. Thank you so much.